So we're going to start our second cell video, and this video is all about cell transport. And we're going to be talking about all the different aspects of cell transport, moving things in and out of the cell. So where does cellular transport happen? Cellular transport happens at the cell membrane for the most part. There is some specialized proteins inside the cell that move things around, but we're going to focus on the transport that happens at the cell membrane. The cell membrane is majority made up of phospholipids. It also has other components, and you can see those here in this picture. We have the phospholipids, and then we have proteins, we have cholesterols, we have glycolipids, we have tons of different things. Things like channel proteins act as like doorways to allow stuff into the cell. Smaller things can just move right in or out of the cell. They can just pass directly through the membrane. Things like glycolipids or some of these embedded or integral proteins act as like name tags so that other cells can recognize this cell as being a member of the body. So lots of components that make up the cell membrane. But I want to focus on the phospholipids because phospholipids are a really interesting molecule. They have two components in a phospholipid. They have what we call a hydrophilic head, and then they have hydrophobic tails. So when we say something is hydrophilic, we mean that it can interact with water. So the hydrophilic heads can actually interact with water because they have a polar component. When we say something is hydrophobic, like the tail part of the phospholipid, it means it cannot interact with water. It cannot associate with water. And so in order to protect these hydrophobic tails so they don't have to come in contact with water, they are sandwiched in the middle of these two layers of phospholipids. And the hydrophilic heads are to the outside of these two layers. And this is how the cell regulates what comes in and out. Like I said, smaller things can pass directly through the membrane, and there are other ways to get materials in. So let's talk a little bit about how things get into the cell or out. The first way is diffusion, and this is very simple. You move material from an area of high concentration to an area of low concentration. Basically, molecules want to even out. They want to be equally spaced from each other. So here we have a ton of green particles here, very few on this side. So all of these green particles are going to move away from each other and equalize in the space. The same thing will happen across a membrane. So here we have some blue dots. They want to equalize, and over time, they will move across and equalize on either side of the membrane. Now, there's another type of diffusion called facilitated diffusion. Works the exact same way as diffusion we just talked about, but this uses proteins to allow the material to move. So here we have a channel protein, and you can see it's allowing these red particles to move straight through. We also have carrier proteins that have a specialized shape that accepts the molecules and then passes them inside. Another way we move material around is with the diffusion of water or osmosis. So when we're talking about osmosis, we're looking at three different things specifically. We're talking about the different types of solutions: hypotonic, isotonic, and hypertonic. I kind of like to remember these because of their prefixes here. So hyper I think of that as somebody being really active, having a lot of energy. And if you look at this solution outside the cell, there's a ton of stuff going on out here. There's lots of solute out here. Inside the cell, we have relatively few solute particles. Isotonic, we have an equal number of solutes in and out. Hypotonic, we have very few solute molecules outside the cell. So let's talk about how the water will move. In cases where osmosis is going to happen, the water has to move because the solute can't. Maybe the solute is too big, maybe we don't have a protein to move it around. Whatever the case may be, the solute doesn't move. So the water has to, in order to equalize on either side of the membrane. The easiest way to remember which way the water is going to move is it's going to move in the direction of the most solute. So in hypertonic solutions, the water is going to leave the cell and go out into the solution. And that's to try and equalize the concentration of solute outside the cell with inside the cell. So the water moves in the direction of the highest solute, and so it's going to leave. Isotonic, the solute concentrations are the same. So the water's just going to move equally back and forth in and out of the cell. Hypotonic, the water is going to move into the cell because that's where we have more solute. So we need to get more water in there to try and equalize the concentrations. Another trick that I use to remember this is hypo reminds me of hippo. And a cell in a hypotonic solution is going to swell up like a hippo. That may not work for you, but it always works for me. Another way we can get material into the cell is through a process called active transport. This is very similar to diffusion. The biggest difference here is that we're going to be using ATP and we're going to be working against the concentration gradient. So we need to move material around, but naturally the molecules are going to want to move to this side of the cell membrane because there's less material out here. But we don't want it out there. We want it inside of the cell. So we use ATP to get these molecules into the cell. One of the places in your body where you might need to use this is in the case of sodium-potassium pumps in your neurons. So what happens is your cells pump sodium and potassium to one side of their cell membrane, and that basically makes it like charges up the neuron to be able to fire. And so very quickly, your neuron can move an electrical signal along its cell body by allowing those ions to move across the membrane. And then your neuron has to reset by pumping them back across. So this has a huge influence on the way cells can operate. And it's very important. But the big thing that I want you to understand right now is that it requires energy, and the energy molecule that we're talking about is ATP, and it works against the concentration gradient. Other ways that we can get material in and out of the cell. Endocytosis is how we use the cell membrane in animal cells to kind of engulf material and then bring it into the cell. So you can see all of these are just ways to pull material into the cell. Phagocytosis is for large chunks like food. Pinocytosis is for fluids. And then, of course, we have receptor mediated, which is just using proteins to activate the process. And then exocytosis is basically the same thing in reverse. We have a vesicle that attaches to the cell membrane, and then we expel contents into the extracellular matrix. And that's exiting the cell and entering the cell. You guys remember those two prefixes.